So from today onwards, I'll be discussing about a new competency that is all about the reproductive physiology. So in today's lectures, I'll be discussing about mainly three different topics. One is human sex determination, then differentiation of human sex and their abnormalities, and finally I will discuss about the various effects of removal of gonads. So I think we have a little information, a basic information regarding how the determination of sex takes place. So let me start with the very basic concept. You can see here, human sex determination is a genetic process. In that particular process, what we see, there is a transfer of a specific gene or chromosome from their parents to the offspring generations. Now, this transfers of that chromosome or sex chromosomes basically depends on a specific process called the fertilization process. Now you can see, human sex determination is a genetic process that occurs at the time of fertilization. It is primarily based on the type of sex chromosomes that is contributed by the parents. Humans have 46 chromosomes in total, including one pair of sex chromosome. Here's it is XX in female and XY in male. So the mother always contributes an X chromosomes through the X, while father contributes either an X chromosome or the Y chromosomes through their sperm. Finally, we could say, if the fertilization occurs between the sperm cell and egg cells, it will give a specific type of zygote, right? So here's, if the fertilizing sperm carries an X chromosome, the resulting zygote will be XX. If it carries a Y chromosome, the zygote will be XY. It means XX means the female offspring, XY means male offspring. Now, ultimately, we could say, based on the information mentioned here, we could say the father's sperms determines the sex of the child. Now let me explain in details. So as I mentioned, if you see the germ cells of a male, it is XY, and germ cell of female, it is XX. So what we see here during the gametogenesis, it is a process that produces a certain type of gametes. So here in this particular case, you can see, in case of female germ cells, it is XX, that is homozygous. And here in case of male germ cells, it is XY, that is heterozygous. So during gametogenesis, the female germ cells produce homozygote, that is XX, and male germ cell produce heterozygote, that is XY. Now, after the formations of that sex gamete, the next event is the fertilization. So, fertilizations of both the egg cell and sperm cells ultimately gives the zygote. So, here you can see the fertilizations between the female sex gamete, that is X, with the male sex gamete is X, it gives XX only. At the same time, when fertilization occurs between female X with the male Y, it gives XY. It means ultimately the fertilizations of both the male sex cell and female sex cells gives either a male offspring or female offspring. That is totally depends on what type of gametes fuse with which one, right? So here in this particular case, there is a formations of offsprings. The distribution is of 50-50, right? Now, here's if we see the role of a certain genes that played during the determination of sex is a specific type of gene. So what is the name of that gene that played an important role in sex determinations? And what is the exact locations of that particular gene? Yes, we know there is a specific type of gene called SRY gene. And that SRY genes is basically located into Y chromosome only, right? It is totally absent in X chromosome. And the determination of sex of an individual is fully depends on the presence of SRY gene. It depends on the Y chromosome only. Sometimes what we see, not only the sex chromosome, not only the SRY gene, there are certain numbers of genes which are not present in the sex chromosome. Instead, they are present in autosome. They also played an important role in the determination process or differentiation process. Now, we will see about the role of SRY gene and the role of Y chromosome in sex determinations. You can see here the structure of Y chromosomes. If you see the structures, it is composed of two arms. One is shorter than other. And the most important thing is, it is composed of around 59 million base pair. It means this Y chromosome contains around this number of nitrogenous base pair. And it is composed of around 150 genes. Out of this 150 genes in Y chromosomes, there is a specific gene that is present in specific segment of Y chromosome, mainly in the short arm of Y chromosome. And the name of that segment or that name of that gene is SRY gene, right? Now, how this SRY gene takes part in the determination of sex? See here, this SRY genes is also known as sex determining region Y. And this SRY genes located on Y chromosomes. And if you see the role of that SRY gene, we can see it takes part in the differentiation process, mainly in a specific period of embryonic development. So what is that period? That period is around 6th to 7th week of embryonic development. The presence of SRY gene initiates the differentiation of bipotential gonad into testis. You can see this is a embryo and its age is around 5 to 6 weeks only. In that particular conditions, we cannot, and in that particular conditions, we cannot say about the sex of that particular embryo. But by the process of differentiations, we can see how the bipotential gonads is either converted into or developed into male sex or female sex. So now question is, what is bipotential gonad? Yes, in this particular embryo, the gonad is bipotential. So bipotential gonads are those which has the potential to develop either into testis or ovary, right? But whether it will develop into testis or ovary, that is totally depends on the presence of that SRY gene, right? Now you can see here, this is a bipotential gonads. If you see the structures of the bipotential gonad, there is two different ducts, and these two different ducts mainly involves with the developments of two different sex, right? The ducts are called Wolffian duct, that is the blue one, and Mullerian duct, that is the pink one. This the Wolffian duct of this bipotential gonad mainly developed into male sex or testis, or it involves with the developments of male internal genitalia. Whereas the Mullerian duct is mainly responsible for the developments of female reproductive organ. Right? Now question is whether the developments of or differentiation of male sex or female sex will occur, that is totally influenced by the Y chromosome, that is totally influenced by the SRY gene. You can see from here, in presence of SRY gene in Y chromosome, it promotes the synthesis or secretion of a certain type of hormone that are called male hormone. So what are those male hormones? One is anti-Mullerian hormones or Mullerian inhibiting substance or MIS, testosterone and dihydrotestosterone. All these three hormones are mainly involved with the developments of both the male internal genitalia as well as male external genitalia, right? And this hormone synthesis or these hormone secretions is totally depends on the differentiations of Wolffian duct only, because Wolffian duct is responsible for the developments of male gonad, right? Whereas in absence of SRY genes, you can see there is a development of female internal genitalia or female reproductive organ, right? Now question is whether only SRY gene involved with the differentiation of bipotential gonad or other genes also takes part. Now we'll see. Here you can see the developing testes then secrete two important substances. As I've already mentioned, it mainly secretes three different hormones. The first one is testosterone from specific cell types, that is the Leydig cells. So what is the role of that testosterone hormone? Yes, it mainly promotes the developments of male internal genitalia from Wolffian duct. You can see this is the Wolffian duct, and that Wolffian duct is developed into male internal genitalia, but this male internal genitalia developments is promoted by the testosterone that is secreted from Leydig cell. On the other hand, another important hormone, that is MIS hormones or anti-Mullerian hormones, that is secreted from Sartorius cells. So what is the role of that anti-Mullerian hormone or MIS? It inhibits or it regresses the Mullerian duct, which is mainly involved with the developments of female gonad. So here you can see which causes regressions of the Mullerian duct. It means the regressions of Mullerian duct means inhibitions of female sex development. Am I clear? So here both the duct played an important role in the differentiation of gonad or differentiation of bipotential gonad. That the differentiations is influenced by the presence of SRY genes, the presence of certain type of hormone like testosterone, anti-Mullerian hormone. Other than this, you can see the testosterone hormones which is converted into dihydrotestosterone, and this conversion is influenced by a specific type of enzyme. The name of that enzyme is 5-alpha reductase. So what is the role of that dihydrotestosterone? The role of that dihydrotestosterone is it promotes the developments of external male genitalia, right? So all these three hormones like testosterone, anti-Mullerian hormone and dihydrotestosterone are the product of testis, and this testis is developed by the process of differentiation from Wolffian duct only. But here in the absence of Y chromosome, absence of Y chromosome means absence of SRY gene. So in absence of that SRY genes, by default there is a development of female reproductive organ, female internal genitalia, right? And at the same time, what we see is in this particular conditions when SRY gene is absent, SRY gene is absent means there is no chances of the formations of testosterone, there is no chances of the synthesis or secretions of MIH. So in absence of that anti-Mullerian hormone or Mullerian inhibiting substance, there is no regressions of Mullerian duct. right? So no regressions of Mullerian duct means the developments of female genitalia. Instead, it regress the Wolffian duct, means it prevents the developments of male gonad, or it prevents the development of testis. Another important point here is the development of external genitalia in female. So, in previous case, what I have seen, the external genitalia of male depends on the availability of dihydrotestosterone, which is produced from testosterone in presence of enzyme alpha reductase, right? So here, as estrogen is absent, so Wolffian duct will not act. No chances of the developments or differentiation of testis, so no formations of testosterone. No testosterone means no productions of dihydrotestosterone. So absence of that dihydrotestosterone ultimately allows the female pathway. Mainly, it promotes the developments of female external genitalia. Now, from that part, we could say human sex determinations is tightly regulated process involving some genetic signal, some gonadal developments, and hormonal influences. But sometimes what we see, you can see some abnormalities either in the genetic signal, you can see abnormalities in the developmental process, and even you can see either hypersecretions or hyposecretions of a certain type of hormones that ultimately promotes the dysgenesis. So these are called abnormalities. Abnormalities in any part of this pathway can lead to disorders of the sex development. So what are the most important disorders that we can see? One is Androgen Insensitivity Syndrome, Turner Syndrome, and Klinefelter Syndrome. Now see, as I mentioned, not only SRY gene, there are some other gene types that also involves in the downstream cascade of SRY gene. And that gene segments are not present in Y chromosome, that gene segments are not present in X chromosome, that gene segments are present in autosome only, right? So now question is, what are those gene segments, how they involve in the downstream cascade of SRY gene or downstream cascade of differentiation process. Here's the one important thing is the TDF. So that is called testis determining factor. The testis determining factors is basically produced in presence of that SRY gene. But when this SRY gene is absent, this testis determining factor or TDF is absent. So now we'll see how this TDF promotes the development of testis and how absence of TDF inhibits alters the gonadal development. Here's, this is embryonic gonad. In these embryonic gonads, there are two different segments. One is the medullary regions and the cortex region. So this medullary region is mainly involved with the male pathway, whereas this cortex region is involved with the female pathway. What? The activity or the functions of both the male and female pathway is influenced by that TDF. You can see here, in presence of TDF, TDF induces the medulla of the embryonic gonad to develop into the testis. Whereas the lack of TDF allows the cortex of the embryonic gonads to develop into ovaries. Right? That means here TDF played an important role. Here the development of ovary occurs or takes place in cortex area, or medulla is developed into the testis. Right? Now, see here, so what are the other genes? What are the other genes that involve with the differentiations of male gonad, and what are the other genes that involve with the differentiations of female gonad? This part is already covered. Yeah? The expressions of that SRY genes produce or encodes a specific protein called DNA binding protein, and this DNA binding proteins triggers the testis development. Up to this is clear. Now, see here, what is the downstream cascade of that SRY gene? This SRY genes actually activates some other genes, mainly the SOX. This SOX is a SRY related HND box gene 9. The activations of this SRY related HND box gene 9 is fully depends on the expression of that SRY gene, expression of TDF, right? So now see, in presence of this SRY, there is activations of SOX9. Once SOX9 gets activated, it activates some other signaling molecule, right? So what are the other signaling molecules? Yes, first you see, it promotes the cell differentiations, maintain its own expressions by a positive feedback loop. You can see it maintains its own expression by a positive feedback loop, and it directly activates the anti-Mullerian hormone gene. So this is the anti-Mullerian hormone gene. The activations of that anti-Mullerian hormone genes leads to the expressions of anti-Mullerian hormone or synthesis of anti-Mullerian hormone, which is required for the development or for the testicular development. Along with this is also activates or promotes the expressions of some other gene segment like fibroblast growth factor 9 as well as prostaglandin D2. Both the fibroblast growth factor 9 and prostaglandin D2 also equally important, though they are autosomal gene. Though AMS is autosomal gene, but they takes part, or SOX is autosomal genes, they takes part in the testicular development. Not only they activates this particular gene, they also, SOX also inhibits the expression of some other genes which is specific for female pathway. So now question is, what are those gene segments that is inhibited by SOX? See, this SOX inhibits WNT4, RSPO1 and FOXL2. They all are the gene segment that is required. The expressions of these gene segments is required for the female pathway. But when SRY gene is present, it activates the SOX. When SOX is present, it promotes the male pathway, but it inhibits the female pathway, right? Now you can see, ultimately it inhibits the ovarian genes like WNT4 and FOXL2. See here, ultimately this SOX involves the differentiation process, like it involves with the expressions of AMH, and expression of AMH produce AMH hormones that causes the regulations of Mullerian duct, means inhibitions of ovarian pathway or female pathway. Along with this, it produces desert hedgehog. It recruits and differentiates the Leydig cell, and Leydig cells is important for the synthesis of testosterone. And PDGF is also important for the overall development of Leydig cell. Now, how this Leydig cells or how this testosterone is involved in this developmental pathway or differentiation pathway? See, in presence of DHS and PDGF, there is a secretion of testosterone, and this secretion starts at around 8 weeks of embryonic developments, and this testosterone maintains the Wolffian duct, and ultimately this Wolffian duct promotes the developments of male internal genitalia, as well as the 5-alpha reductase converts the testosterone into DSD that involves the developments of external genitalia of male sex. Now, in case of female, as I have already mentioned, these are the most important gene that promotes the female pathway, and the activity of this gene in presence of SRY are inhibited by SOX9. Here, in female pathway, you can see the role of WNT4, role of RSPO1 and FOXL2. See here, this WNT4 activates another important signaling molecules, that is beta-catenin, and ultimately in presence of that beta-catenin suppress the SOX9. So suppressions of that SOX9 means suppressions of male pathway. Instead, it promotes the female pathway, right? So ultimately beta-catenin suppresses the SOX9 and enters the inhibitions of testis formations. No testis formations means no testosterone, no DSD, no AMH, no male pathway, right? And on the other hand, RSP involves some other way. It enhances the activity of WNT4 and beta-catenin signaling cascade, right? And FXL2 act as a transcription factors, which is essential for the granulosa cell development. And this FXL2 also involves the suppressions of the expression of SOX9. It ultimately promotes the female pathway. So these are certain types of gene which are not present in sex chromosome. Instead, they are present in autosome. They also equally important in the differentiation process. Now see, as I've already mentioned, regarding some abnormalities. So what are the most important abnormalities that is involved with the differentiation of gonad? And what are the most important factors that involved with that affects the differentiation process? Yes, there are certain factors and there are some causes of that abnormalities, mainly disorders of sexual differentiations. So what are these? Some are chromosomal abnormalities. Some cases, what we see, either there is more numbers of chromosomes instead of normal chromosome numbers, as well as in some cases what we see, there is a structural abnormality in chromosomes. Both the number abnormality and structure abnormality affects the differentiation of gonad. So most important chromosomal abnormality that we can see in the subjects who are having Turner syndrome and Klinefelter syndrome. So in one case, what you see, the chromosome number is 45 plus X0. In another case, what you see, the chromosome number is 46 XXY. So instead of one X chromosomes, there is more than one X chromosomes as a sex chromosome. So another important part of this disorders is gonadal dysgenesis. So it means it is improper developments of the gonads, both the male gonad and the female gonad. Sometimes the hormonal imbalance also is responsible for the developments of some disorders during sexual differentiations. These are called excessive or deficient level of sex hormones, such as that is found in congenital adrenal hyperplasia. And in some cases, we can see androgen insensitivity syndromes, a condition so the body does not respond properly to male hormones. And the absence of or deficiency of 5-alpha reductase is also responsible for the disorders. In these particular conditions, where the body cannot convert testosterone into dihydrotestosterone, so which is very crucial for the development of external genitalia. And the mutations or genetic mutations also equally important. That mutations leads to the disorders in the sex development. Now, these are some informations like the three major classes of DSD. In this particular conditions, you can see the chromosome number changes, right? And here also you can see, though the karyotype C is normal karyotypes, but due to the deficiency of this particular enzyme, there is a abnormality in the developments of external genitalia in male sex. Though it shows the genetic contribution shows the normal distributions, but due to the insufficient of this particular enzymes, it affects the overall external genitalia developments. Here also, in case of this 46XX DSD, the chromosome numbers or karyotype is normals, but if you see the external features, the external features is not normal according to the chromosomal distribution. So these are the most important disorders that are related with the differentiation of sex or differentiation of gonads. Now comes to the next part. Yes, Now we'll start with the effects of removal of both the male gonad and female gonad. Yes, we'll see there are numbers of effects that may occur due to the removal of that gonads. So what are the effects that is related to the removal of testes? You can see the loss of testosterone secretions. If there is a removal of testis, so the most important effect that you can see is the loss of testosterone hormone secretions, and this testosterone secretion loss ultimately affects the overall development. Like it reduces the development of secondary sexual characteristics. It also reduces the libido as well as sexual functions, and even in absence of these testosterone hormones, there is a high chances of the loss of fertility, right? So these are the most important effect of the removal of testes that is basically occurs in the primary genital organs of male sex. At the same time, due to the removal of that testis, it also affects some accessory sex organs, like you can see the atrophy, right? So atrophy in different parts of the accessory sex organ. So these are the most important effects of removal of testes. Other than this, you can see the effects of this testis also affects the skeletal parts of bone. There is a decrease in the bone mineral density. So decrease in bone mineral density leads to high chances of osteoporosis in subject. This is another important effect of removal of testis. So what are the behavioral effects? Reduced level of aggressions, drive as well as energy, and frequent mood changes, right? Along with the depressions and lethargy. So what are the metabolic effects of testis removal? So there is these are some metabolic effects, like high chances of increase of body fat and decrease of lean fat mass, and at the same time you can see the possibilities of the development of mild anemia, and it is mainly due to the decrease in the synthesis of erythropoietin hormone, right, or erythropoietin stimulations. Now what are the other parts? So what we see in the removal of ovaries? See here, it means, as we know, the ovary is the most important part for the synthesis or secretions of female sex hormones, estrogen and progesterone. But removal of that ovaries means loss of estrogen and progesterone secretions. So that ultimately affects the different biological events. The mostly is the menstrual cycles is affected by in absence of both the estrogen, means lack of estrogen and progesterone. Along with this, the persons can enter into the premenopausal, premature menopausal phase, right? This premature menopause is directly related to the development of various type of metabolic disorders. It also affects the secondary sexual characteristics, like it regresses the mild, it is a regressions process, means mainly you can see the effects of that removal of ovary in the post-pubertal phase, right? And even the development of breast is also can be affected due to the removal of that ovary. So along with this, we can see the reproductive effects as well as the effects in the skeletal parts of the body. So what is the effects in skeletal part? Again, there is high chances of the development of osteoporosis in female. So because there is due to the loss of that estrogen progesterone, there is a loss of bone formations, ultimately promotes the osteoporotic events. So these are the most important effect that we can see in both the male and female if there is a removal of testis and ovaries. Now this is what I wanted to discuss in today's lectures. I hope up to this now it is a little bit clear to you people. Now it is open for discussions. If you have any doubts, If you have any doubts, queries, you can ask.